bi bi proteins, the major light-harvesting pigments of the cyanobacteria. It is thus not surprising that the red algae chloroplast and endosymbiont of quartz shows close phylogenetic affinities to the cyanobacteria. Their host cell, by contrast, forms a distinct line of eukarya. Dinoflagellates are flagellated primary marine algae that are closely phylogenetic relatives of ciliated protozoa. Some dinoflagellates are free-living, while others live in symbiotic existence with animals that make up coral reefs, obtaining a sheltered and protected habitat in exchange for supplying photosynthetic fixed carbon as a food source for the reef. Toxic dinoflagellates. Dinoflagellates such as Gonalax, Gonialax occasionally grows in dense suspensions called blooms that are bright red in color from the xylophiles present as accessory pigments in the organism. These blooms are classic red tides that occasionally develop in warm, easily polluted coastal waters that are associated with fish kills and poisoning in humans following consumption of contaminated water or shellfish. Toxicity occurs because of the potent neurotoxin produced by Gonalax that is sufficiently toxic to kill fish at nanogram levels. Physteria is another genus of toxic dinoflagellates. Although capable of photosynthesis, this organism is notable as a fish pathogen and association pathogen of humans. Toxic spores infect fish and eventually kill them from neurotoxins that affect movement and destroy skin. Lesions on areas of the fish where the skin has been removed, and this allows the growth of opportunist bacterial pathogens. Fish killed by P. Isidia infection are then fed upon by the amoeboid from Physteria, which is itself harmful saprophyte. All told, over 20 different stages are known in the life cycle as Physteria, only one of which, the toxic spores, seems capable of causing disease. Appears that the presence of fish in some way triggers formation of the toxic stage in the Physteria life cycle, and thus fish are the main target of infection. Cause massive fish kills. For example, over 1 billion fish were killed by the Physteria outbreak, and the new Esteria, North Carolina in 1991, and other outbreaks along the eastern coast of the United States have been suspected. Humans can also be harmed by Pisidia, and those inflicted display a variety of seemingly unrelated symptoms. They include respiratory problems, headaches, aching joints, muscles, disorientation, and memory loss. I mentioned previously another toxic dinoflagellates, such as Gonalaxis. Human disease occurs when people come into contact with contaminated water or fish, especially shellfish. But this is not true of Physteria. Instead, the organism produces a volatile neurotoxin that can cause symptoms even when no direct contact with infected material has occurred. Contaminated water is also a vehicle for transmitting disease. Although the shellfish harvested from Physteria-contaminated waters apparently do not transmit disease, Physteria is thus very interested in complex algae, whose ecology may be tied much more to a lifestyle as fish pathogen than as a phototroph. Several characteristics are used to classify algae, including several pigments, energy, metabolism, reserve polymers. Several characteristics are used to classify algae, including the nature of the chlorophyll, the presence of carbon reserve polymers produce, for example, starch and several starch derivatives, the cell wall structure, and the type motility. All algae contain chlorophylls. Some, however, also contain other chlorophylls that differ in minor ways from chlorophyll. The presence of these additional chlorophylls is characteristic of particular algae groups. The distribution of chlorophylls and other photosynthetic pigments in algae is summarized. All algae carry out oxygen involving oxygenic photosynthesis using H2O as an electron donor. In addition, some algae carrying out photosynthesis without yielding oxygen using H2 as an electron donor. Many algae are oblique phototrophs and are thus unable to grow in darkness on organic carbon compounds. However, some species can grow chemoorganotrophically and catabolize simple sugars or organic acids in the dark. One of organic compounds most widely used by algae is acetate, which can be used as a sole carbon in energy. Source: by many flagellates and chlorophytes. In addition, some algae can assimilate simple organic compounds in the light photoheterotrophy and section, but cannot grow on them as the sole energy sources. One of the key characteristics used in the classification of algae groups is the nature of the reserve polymer synthesized as a result of photosynthesis. Algae of the division Chlorophyta produce starch, 4-linked glucose in the form very similar to that of higher plants. By contrast, algae of the other groups produce a variety of reserve substances, some polymeric and some of free monomers, and the major ones are listed. Cell walls of algae. Algae show considerable diversity in the structure and chemistry of their cell walls. In many cases, the cell wall is composed of a network of cellulose fibrils, but it is usually modified by addition of other polysaccharides, such as pectin, xylins, mannins, alginic acids, fucinic acid, and some algae. The wall is additionally strengthened by the deposition of calcium carbonate. These forms are often called calcareous or coralline. Coral-like algae, sometimes chitin, a polymer of N-acetylglucosamine, is also present in the cell wall. In euglenoids, the cell wall is absent. In diatoms, the cell wall is composed of silica, to which protein and polysaccharides are added. Even after the cell dies and the organic materials have disappeared, the internal structure, called the frustum, often remains, showing that the siliceous component is indeed responsible for the rigidity of the cell. Because of the extreme resistance to decay of these diatom frustums, they remain intact for long periods of time and constitute some of the best algae fossils ever found. From this excellent fossil record, it is known that diatoms first appeared on Earth about 200 million years ago. Diatoms typically show some form of symmetry in the morphology of their frustums. Examples of symmetry include pinnate, having similar parts arranged on opposite sides of the axis, and radial. Algae cell walls contain spores some 3 to 5 nanometers wide that are large enough to pass only low molecular weight substances such as water or inorganic ions, gases, and other small nutrient molecules needed for metabolism and growth. Phagocytic activities are thus not possible, and in this regard, algae differ from the activity phagocytic protozoa and slime molds. Motility and ecology of algae. A number of algae are motile usually because of flagella cilia do not occur in algae. Simple flagellate forms, such as Euglena, usually have a single polar flagellum, whereas flagellated representatives of the Chlorophyta have either 2 or 4 polar flagella. Dinoflagellates have 2 flagella of different lengths and wide different points of insertion into the cell. The transverse flagellum is attached laterally, whereas the longitudinal flagellum originates from the lateral groove of the cell and extends lengthwise. In many cases, algae are non-motile in the vegetative state and form motile gametes only during sexual reproduction. Algae abound in nature in aquatic habitats, both freshwater and marines. Algae are also found in artificial aquatic habitats like fish tanks and swimming pools, and in temporary pools of water formed from rainwater runoff. Algae are also common in most soils, while a few species thrive in dry and even extremely dry soils where the water potential can be very low. Algae are often the dominant or sole phototrophic microorganisms in acidic habitats as well. Below pH, cyanobacteria are absent and several algae species flourish. Some algae species have been found growing inside rocks. These endolithic means inside organisms exist in porous rocks, for example those containing quartz, and are usually found in layers near the surface. Endolithic prototrophic communities are most common in dry environments such as deserts, or cold, dry environments like Antarctica. For example, in Antarctica dry valleys, where temperatures and humidity are very low, life with a rock has its advantages. Rocks are heated by the sun, and water from snow melt can be absorbed and retained for relatively long periods, supplying needed moisture for growth. Moreover, water absorbed by a porous rock makes the rock more transparent, thus supplying more light to the algae layers. A wide variety of phototrophs can form endolithic communities, including cyanobacteria, a various green algae. In addition to free-living phototrophs, green algae and cyanobacteria coexist with fungi and endolithic lichens communities. For discussion of lichen symbiosis, metabolism and growth of these internal rock communities solely weathers the rock, allowing gaps to develop where water can enter, freeze, and thaw, and eventually crack the rock, producing new habitats for microbial colonization. Well, this is the end of chapter 14, and I would like to show you my new jams that my mom bought me for Christmas. Aren't they beautiful? Red and black. They're really, really nice. I decided to wear them today for my video because today is Christmas, and it'd be nice to get out of the lab suit, you know what I mean? Well, thank you, and have a great night. This has been a long session, so good night.